Hey there, how are you?
I'm good. How are you?
I'm good. How's it going?
Good.
Uh-huh.
Test is nearing. It's around the corner. Saturday.
Yep. So, yeah, tell me about how you're feeling. What have you been doing, I guess, since then?
So yesterday I did the whole biochem section bank.
Yep.
It was useful to just get that practice and I mean I did it basically almost all in one sitting, so it was also good for stamina.
Yeah. So you did that passage about like fiber being turned into the short chain fatty acids. Good.
Yeah.
And
Yeah. And yeah, since I've just been doing a lot of like Anki cards,
yep,
yeah.
got it. So yeah, would what would you like to talk about? So oh yeah, I forgot. So I think, did you say last time that like timing you were having some issues with in one of the sections?
Yeah, well, it was a lot better like since like uh
Hmm.
when I got took like the AAMC test, like timing was a lot better.
Yep.
But I was still like struggling against
Hmm.
timing for it's really just like the chem phys or biochem sections.
Yeah. Did we talk about timing strategies before?
Yeah, I think we did. I mean it's helped a lot. But
What did we talk about exactly? Like what did I?
like the three checkpoints.
Oh yes, got it. Okay.
Yeah. But it's just
Hmm.
yeah, I guess I'm not supposed to have much time at the end left.
Yeah.
It's how it's designed, right?
Yeah. So I mean, what you can do, or yeah, what I also say is for any choice that you're not 100% certain of, flag it. But also, if any question is taking too long, right, you want to make a guess and flag it. And the idea is that if you stick to this, like I say, like not to spend more than like an hour and sorry, a minute and a half per question. Of course, you're not going to be able to time, each question, like, how long it takes, but that's generally what you want. And if you do that, like if you are disciplined in that and that if a question's starting to take longer than like nearly 2 minutes, then guess, flag, move on. If you do that without letting any question take too long, you will have some time at the end where you will have seen all of the questions by that point, and then you can hit like review flagged and then go through the flagged ones. Um, so, but also like, yeah, have you like I guess come up with like your list of things to write on like the scrap paper when you start the test?
Funny, because I definitely did, and then I didn't even use it when I took the test.
Yeah. I mean,
I don't know if I really should, because I don't think I will end up using it. Yeah.
yeah. I mean, like, a lot of times that's the case. But how do you think it makes you feel in terms of like confidence? Like, do you feel like when you do that, when you write the stuff like on that scrap, do you feel like you're entering the test with more confidence? Do you feel that, or is it not?
Um, well, I don't know. I've only practiced it once doing
Yeah.
that, but yeah. I mean, I feel like the way that I learned the formulas,
Huh?
like I made my own Anki cards and they were like all always just scrambled. I
Yeah.
kind of got used to them, and like not writing them down. So like I'm not like dependent on having them written down.
Yep.
Um, I thought it would be useful for the units, and then I realized like, oh, like trying to figure out like it's gonna take too much time.
Uh-huh.
Um, so I honestly don't think like that I would use it if I did it on the real test.
Uh-huh.
But I might write some things down. I don't know. I'm undecided yet.
Yeah. So, because yeah, the way that I see it is that you can either get those 15 minutes like as essentially free time for you to uh use if you want, but if you don't want to use it, then you won't have it. But what I would do is like I think I told you like on the way to the test, like I would just be blasting through as much Anki as possible, and certain things will be in my short-term memory that I want to, like, the second I get the ability to, or the chance to write it down, I write it down, and it could be a whatever that I would write down whatever I did not feel comfortable in knowing verbatim. So like the optics table, something like that, or maybe like the structure, like if maybe like, you know, the structure of a certain amino acid, or, you know, things that you like can brain dump onto the sheet of paper that you would forget otherwise, that are just, you know, in your short-term memory. Because yeah, like when I did that, like I ended up not really having to use it either, but it gave me a little bit more peace of mind, and a little bit more feeling of control, being doing that. So yeah, it's up to you. Um, but yeah, okay. What yeah, what type of things would you want to work on?
Um, well, first it's like just a question. So I've sort of like so I hadn't been I guess I have a lot of Anki cards to do, and I'm like trying to see like what's my best approach like the coming days, like today, and I've sort of been like switching around with them. Because originally I was like, let me just do a lot of like biochem ones. And then I also don't want to like forget about like psych social, although I'm scoring really well in it. So it's like, but it's a lot of cards. I don't think I can realistically get through all of them. Yeah. So I just don't know which would you recommend prioritizing?
Yeah, so with Anki, I never really expect, you know, myself or anyone to really like finish it. It's just like something that you do like each day, to just help things stay into your, help facilitate things going from your short-term memory to your long-term memory. So yeah, so I wouldn't worry about like how many cards you have left or how many cards you're going over, as long as you're going over the cards that, you know, that you don't know, because that's how like it works. But for, in terms of which cards to focus on, like yeah, the psych soc always, because that's this, you know, like aside from like BB, where they can just ask you like straight up like what's after fumarate or something like that. Like that's, like you need to know the content for that. Same thing with psych soc, except even more of it would be like that, where you just have to know it. And I feel like since the psych soc section, like if you just look up percentiles and average scores, like, you know, of everyone who took the MCAT, the psych soc section is the highest. And that works two ways. Like if you know the material, which is what pre-meds are known for, they're known for, you know, having a lot of material memorized and stuff. If you do that, then you'll do well. But if you don't do that, it's hard to use like critical thinking in that section. So even though you're doing well in psych soc, I always hear from students who take the test like that they always say something like, you know, psych soc was had a bunch of stuff that I didn't really learn about or something like that. So I think with psych soc, they're always going to be throwing new terms at you. And it's hard to get a grasp on how many different types of topics they can ask you in psych soc. So I think that doing the deck, I think you mentioned like the Pancow deck. Like yeah, so I hear that that's a really good deck for psych soc. So so okay, yeah. So I would say just in areas that you feel where your content knowledge can be improved, to do that.
Okay. Yeah.
Mhm.
All right. Yeah. So there's a question that I keep getting wrong.
Okay.
And it's especially because I've seen it in like different circumstances. Like the palindromic sequence questions. with like restriction enzymes and like sticky ends.
Mhm.
So I can pull up a question on UWorld that I did today. This, maybe it's this. Yeah, but not this. So, yeah, this one.
Okay.
Um, yeah.
So why don't you, is this something that we need the passage for?
I think you need this table with like the restriction sites. Well, I'm unsure, but yeah. So we have a gene
Uh-huh.
and we're digesting it with this one and XO and this one. And well, they give us the like plasmid CLDNK.
All right, let me take a quick look at this passage. It seems like a lot's going on. Um,
Okay.
so let's see. Okay, in the central nervous system, myelin, produced by oligodendrocytes, glial cells, functions as an insulating sheath surrounding certain nerve fibers. To identify candidate genes involved in the myelination process, researchers collected oligodendrocytes from zebrafish and purified the messenger RNA, the mRNA transcripts expressed by these glial cells. The mRNA was converted into complementary DNA, cDNA, by reverse transcriptase, and the cDNA was fluorescently labeled and addressed for hybridization on a microarray. The expression of mRNA in other zebrafish cells was similarly measured. Okay, what's being told here?
Um, so they have like oligodendrocytes from zebrafish, and they also collected like other cells from zebrafish, and they want to know like which transcripts are like differently expressed between these two different cell populations in order to identify like which ones are involved potentially with myelination.
Excellent. Okay. All right. Next, transcripts detected in zebrafish oligodendrocytes at levels greater than 3 times those found in other cells were selected as candidate myelination genes. Okay, so they identified mRNA transcripts uh that okay, the mRNA transcripts that they found in the oligodendrocytes of zebrafish were 3 times as much as the regular cells of the zebrafish. And so the ones that have 3 times more, um,
Or like their hits kind of in the screen.
yeah, that's what they're gonna like investigate in terms of if they're gonna be genes responsible for myelination. So okay, some of the detected genes, including PLP1A, SOX10, and MBP, were previously known to be specific to oligodendrocytes, validating the procedure. Okay. The cDNA of newly identified candidates was amplified by PCR. Whenever you see PCR, by the way, there's been a bunch of buzzwords so far, but yeah, whenever you see PCR, you know, start thinking about things like, well, actually, I'll just, well, yes, think about how restriction enzymes play a role in that. So they might ask you something like that. Okay, the ends were digested with restriction enzymes EcoRI and ZOL. The PSK2 vector shown in figure 1 was then digested with EcoRI and ZOL, and genes were ligated into the multiple cloning site, MCS. The positions of various restriction sites in the MCS are shown in figure 1. All right, so this is a super, super dense passage, like straight up. Very, very hard to see, like, I guess what's going on because there's just so many acronyms, but you're good. You've had, practice in this. So, okay, we see plasmid here, what kind of things do you see in this plasmid?
Um, well, AMP resistance, which is probably like ampicillin,
Mhm.
like an antibiotic resistance gene. Laxi. I don't know what it is. I mean, reminds me of the lac operum, but probably not. Um, MCS, unsure, and Ori, unsure. But I'm told the amount of base pairs, so sort of like the overall size, great guess. Is that
Good.
it?
Yeah. So this is essentially just showing us um the different site restriction sites in the multiple cloning site. Um and yeah, and that's essentially what we get to see then in figure 2.
Mm,
Yep. So like, yeah, if you scroll up to like where we just have figure 2 on the bottom.
figure one or
Oh,
which?
just where you were, yeah, right here. So we see that in this MCS multiple cloning site, uh that there's the stuff in figure 2.
Yeah.
Like, yeah, like this MCS over here is this thing in figure 2.
Yep. I
Okay.
see.
So we see essentially, it looks like a box, but it's like one strand 5 to 3, and then the other strand 3 to 5, and then we have I guess these regions where the particular restriction enzymes would, cut. So XFAL is at that beginning region. BAM HI is later on, 12 base pairs like over, and then yeah, it continues going that way. So okay, we see that stuff. And let's continue. So okay, table 1 shows the functional parameters of several restriction enzymes. So we have, oh, okay. So we have the different restriction enzymes and then the site at which they would make the nick, right? Like the cut. Um, so yeah, so we have that. So like for instance, like this one would be like that as a fragment, right? And then this would be the other fragment, right?
Oh, I didn't know it was like that.
Yeah, right. Because if you so if I drew this, right, GGATCC, and then the same thing, like complementary on the bottom, CCTA GG, right?
Yeah.
So we can see that this is a palindromic sequence, right? Because so a palindromic sequence is like, well, a palindrome is a word that is the same forward as it is backward.
Yeah.
Um, Palindromic sequences in this context is going to involve like something that goes in one direction, like that's complementary to the palindrome of it. So
I
like
see.
like race car, we know as a palindrome, right?
Mm-hmm. Yeah.
So underneath it, I could draw like the same thing but backwards, right? Race car, right? So regular palindromes, it's the word itself that's the same forward and backwards, but palindromic sequences, the forward part will be one strand and the backward part will be the other strand, and they'll be complementary.
Um, the what confused me here is when you like separated the cuts into like those two sections,
Yep.
because I thought it would just be like between G and C and between like G and G on the bottom instead of those actual like two
Mhm.
yeah.
Yeah. So that is what you have happened. So basically, this there's like a cut here after the first G, and there's a cut here, right?
Yeah.
Those are the only things that are cut. Now, that doesn't, like, that's, yeah, those are the only things that are cut. And but that doesn't mean that the two strands are like separating. The strands are still complementarily attached, right? So if you just made these cuts here, right, what you would end up with is like something like this, right?
Yeah.
And then something like that. Because you're just cutting like in between like the two guanines of one strand here, and then you're doing it between the guanines on the other strand underneath. That's all you're cutting. So if that's all you're cutting, that means that the stuff here, like this, that stuff will still be bound, right? So
Yeah.
yeah, so this essentially means that you have this as one of the fragments, and then the other one is this as that other fragment. And since they have these overhangs, they're called, that means that they can bond to each other again. Um, because of the fragments being we can also call them sticky ends, right? As opposed to if, you know, if it was something like that would just be like a blunt end, which would just be, you know, maybe something like this and like this. They don't really fit in like in that jigsaw puzzle way. So yeah, so when you think about restriction enzymes, think about it that way. Think about, you know, you're just making a cut in, like, you're actually cutting through like the phosphate backbone as well, but you're leaving the complementarity intact, right? So okay. And then we see some stuff with, okay, pH, optimal, enzyme conditions, and like, I guess, NaCl, concentration, and then this other thing, but we don't really know about that yet. But what we can see, so okay, what would the sticky ends look like for, what is that, C-Lal, I think?
I see. So it would be like CGAT and then TA would form one fragment. And then the other would be like ATTAGC.
Mm-hmm. Yeah, good. Um, and yeah, and essentially we see that for all the other stuff, right?
Yeah.
And different other conditions, but we don't really know how to deal with that yet. But okay, so we see all these restriction enzymes and specifically where they make their cuts. So okay, then it says to confirm that the candidate genes originated in oligodendrocytes, complementary RNA, cRNA strands, were synthesized from the cloned plasmids and hybridized to fixed zebrafish sections. One of the genes that localized to oligodendrocytes, known as CLDNK, produces a 915 base pair mRNA transcript from 2 exons, and the CLDNK gene is involved in tight junction formation and may be required for myelin sheath development around axons. Okay, so yeah, so much going on here. So right away, like if you were to see this on the test, like I don't know if I said this to you before, but when you like read it, you're going to be looking out for those buzzwords, and when you approach the figures and the tables and stuff like that, you're not going to like, I would say like 15 seconds or so to spend on the figures in the table and stuff, because the reality um is that if they ask you questions based on the figures and tables, you can just look back at the figures and tables. But a lot of times they won't ask you, or they'll ask you something that's kind of easy. Um, and that's when you would go back. I usually say don't go back to the passage for different things, but that's when you would go back to the passage to look at it. Because if we were to analyze table one without any context, like in the sense of like no question that they're asking us yet, we don't really know like what to really pay attention to, because I mean, like, we have like something here, something here, something here, something here, and then 0, 0 for there. We don't know what that really means, right? But it's okay that we don't know what it really means, because we don't need to know until they ask us something about it. So that's just how I would say to go over passages in general. Now, okay, so let's see. So they're talking about, okay, the question stem is asking which of the following diagrams shows the structure of the cloned CL-DNK gene after digestion with EcoRI and ZOL. So what do we do here?
I guess we would go to like EcoRI and Zoll and see like what the sticky hands, sticky hands, yeah, that's what they call, would be.
Uh-huh.
Um, yeah. So I guess we have like four possibilities, and like a combination that has like two of those would be correct.
Mhm.
Perhaps.
Yep.
Uh,
So, did this give us, can I see what the above a little bit? Figure two, I mean. Okay, so we have these different sites here.
oh, I see. Like the 5 to 3. So we kind of know what is it that like EcoRI should be on like the left,
Uh-huh.
perhaps, and Zoll to the right.
Uh-huh. So yeah, so go from there. So why don't you start off with looking at like the choices, and then I guess identifying where the nicks would be from the table
The okay, like going from like option A
one. Mm-hmm.
for like identifying like which enzyme was used.
Mm-hmm.
Okay. Um, I'm trouble finding this one. G This is almost this one, but the cut is not there for A, so I can't find one that fits for that side. Uh, okay, and like Zoll would be for this side.
Yeah, so what we want to be looking at is for the eco R, oops, RI, we should see like the restriction site will be like GAATTC, and for the ZOL, it should be CTCGAG, right?
Yeah.
All right, and um yeah, and we're being told that the gene here is going to be well, when we use these restriction enzymes, right, we want to think, okay, where will it cut this, and where and which, I guess, sticky end would be like on which side, right?
Yeah.
Okay. So,
I think I got it. And I can see it
uh-huh.
for the answer. Like, what else should I know for this? Because I mean, I think for the section bank, I got a question wrong, which was like they used restriction. Sorry, they were using Southern blot. And they were identifying a missense mutation. And I had to know that like for a Southern blot, they would be using like restriction enzymes.
Mhm.
And so like the missense mutation would be like, and if it was identified through a Southern blot, then my mutation had to be in a palindromic sequence,
So
which was very,
yeah,
yeah.
so anytime you're dealing with like a blot like that, you're going to open up like a gene, or open up like some DNA. So um restriction enzymes would be used for those types of you know experiments like that.
Yeah.
Um but it can also be used like in this context, I guess, in like I guess inputting genes into like a plasmid and stuff, and having the plasmid connect again. So we're if we're trying to I guess isolate that CLDNK um gene, um we would want like one of the ends to be what you would have if eco or I were to do it, and then the other end where the Zol would do it. So so essentially what we can do is we can draw like our sticky end, so like G A T T C, and then they're here. Oops. Then that. And then for this one, it would be GA, wait, yeah, GAG, CTC, and then they cut here and here. So you would get that and that, right?
Yep.
Now we just want to find if we can see that in um in like our choices.
Yeah.
So you could see that. So this one has CGAGCT, right? And then this one is AATTCG, right?
Yep.
Right, because we're going to be, yeah, so those are going to be the sticky ends that are going to be flanking this gene. So, and you see that, well, this looks like, I'm going to just make that so it's a C. So we see that we're going to need to see these types of fragments, right? And we don't really see that in like, well, A on the left here, whoops. Yeah, A on the left here. That is like, okay, tell me about that. Like, wouldn't that like work? Because it has GAATTC, and from here we see that.
For the cut. That was
Exactly. So A, it's not cut, right? This thing, right? And the same thing with this. GAGCTC. That's like a palindromic sequence for the Zol. And it's not cut, right? The if it didn't have that C, right? And if this didn't have this G, that'll be something that works.
Yeah.
Right. So, and then we see that in B, where we actually see these sticky ends. And then in C, it seems like we have the correct one on the left, but on the right, we don't have the correct, like this one does not have the correct sticky end for the Zol,
Yeah.
right? But like this is the correct one for Eco RI, right?
Yeah.
And then this one here is also like not cut yet. And then this one on the bottom here, I mean, sorry. And then this one over here, that is, yeah, I don't even see that.
It's X ball.
Oh yeah, yeah. So that's not going to be it. So yeah, what you want to do is you want to, so yeah, this, I guess now you know that those arrows and stuff, like the sites where it's cut, that will produce those sticky ends. And it will have to be at palindromic sequences, because if it is not in palindromic sequence, it's not going to be able to bind back together.
Match. Okay.
Mhm.
That makes sense. Yeah.
Yep.
Sounds good. I have another question from this time from the section bank. It's this question. So it's asking like it's a discrete. Which event is better? Mediated by a ligand-gated ion channel.
Mhm.
So yeah.
Okay. So why don't you walk me through these choices and tell me about what um yeah, tell me like what they're describing, I guess.
Yeah. Release of calcium from the sarcoplasmic reticulum of a muscle fiber to initiate muscle contraction. So it's like yeah. So I am thinking of like acetylcholine, like binding, and I mean I'm not sure exactly what like the like which receptor is responsible for which, and they're all on basically muscle contraction, um, which is what to me made it very tricky. Um, yeah.
Okay, um so you talk about acetylcholine, like tell me like the whole process from the beginning to the end.
Yeah, so acetylcholine is like the ligand for like I guess an acetylcholine receptor. And my thought is like when this happens, it causes depolarization and
Let's take a step back. Let's talk about calcium being released from the sarcoplasmic reticulum.
um calcium is released. I guess it has to be released from a channel.
So
Uh yeah.
so T tubules, right?
Yes.
Like let's say something like this in the sarcoplasmic reticulum, and let's say this is the muscle, um this is uh the neuron, and so this is the neuromuscular junction. So
Mm-hmm.
calcium is sequestered in the sarcoplasmic reticulum, and they can be released out here, and they can be taken up by the neuron. And when the calcium is taken up by the neuron, it will cause acetylcholine to exit.
Okay. I see.
right? And I'll draw this a little bigger here. And that acetylcholine will bind to an acetylcholine receptor that's on that muscle or the motor end plate. And when it binds to that, it causes that depolarization to continue.
So
Right.
what causes the release of calcium? Is that support that's
Okay, so I just want to say before that, in order for this depolarization to continue on to that muscle, what happens in depolarization?
the membrane passes threshold. I'm not sure what you're referring to exactly.
So tell me what depolarization is.
It means the membrane becoming more positive.
All
So
right.
normally it's like at a resting potential around like -75
So how does
millivolts.
more
Huh?
so how does it become more positive?
Like the voltage gated sodium channels will open.
Okay,
Mhm.
which causes.
The action potential rise.
So but if
The
the channel opens,
the depolarization. Yeah.
so like if the channel opens, what
Mhm.
happens with that sodium though?
The sodium goes in.
Goes in, right?
Mhm.
Okay. All right. So let's see. So let's see. So A says release of the calcium from the sarcoplasmic reticulum of a muscle fiber to initiate muscle contraction. That'll be something that will happen after the after the muscle gets that action potential, that depolarization that will then help the calcium can then, so the calcium initially will cause the acetylcholine to be released. And let's say that does its thing, um, which will cause the action potential to move on into the muscle. And then the calcium can actually bind to the, I think it's a troponin, which will help cause a kind of shift in the conformation, exposing the sites uh, that myosin can bind to. Um, but okay, I guess that doesn't help us realize. Well, okay, so I was gonna say it doesn't help us realize if choice A is ligand gated or like voltage gated, but if we don't know like why it's released, it's a good chance that it's not ligand gated. So let's just kind of like table A for now. Let's think about what is ligand gated here. So I mean, like, all right, let's just go look at B. So B says influx of sodium across the axon membrane of a somatic neuron during action potential propagation. What is that describing?
Voltage gated.
And like tell me more. Like what is it describing, I guess, like in terms of like for instance, like I wrote like I drew like the neuromuscular junction, right? Um, but B would be between two neurons, right? Like for a synapse.
Yeah.
So and that's just action potential going from one neuron to another. And that's happening through like without ligands being needed, right? It's happening because of that voltage, uh, well, voltage gated channels letting the, because, okay, so you mentioned like if the action or what was it called? Like there's a threshold at the resting potential, and if it goes above a certain threshold, it can start that depolarization. And that means that the depolarization, which means the sodium channels are opened, and since there's a higher concentration of sodium outside the cell compared to the inside, sodium influx comes in, right? Now, that's a channel that opened up. The sodium channel is opened up, but it's opened up because your threshold potential is, you're going over that, right? So a change in voltage is what causes that depolarization action potential generation in neurons, right?
Yep.
So B is just talking about action potential propagation, which is making, neurons like depolarized, which happens from sodium influx, but it's not going to be ligand gated because we know from what we know about, the depolarization and repolarization and stuff like that, that this is not dependent on any type of ligand binding to anything, right? Now if we look at D, it says re-entry of calcium back into the sarcoplasmic reticulum of a muscle fiber to end the muscle contraction. So that'll just be like, this going back in, right? But in all of these, right, the only thing that is ligand gated, right, is when the acetylcholine neurotransmitter is released into that neuromuscular junction, and it binds as a ligand to a ligand gated sodium channel, which is what this is. And that opens up that sodium channel, allowing the sodium influx, causing that action potential to essentially cross like from the neuron to the muscle itself.
I see. I'm still confused with the order of things.
Uh-huh.
So yeah, so acetylcholine, like
Uh-huh.
the acetylcholine will bind and it will cause the depolarization of and yeah, I'm so confused of like what's leading to the calcium release from the sarcoplasmic reticulum.
So I think that will just be something, I guess, if you are going to yeah, I'm trying to think about if you are okay, so it's at the very least we can know that only one of these choices will be ligand gated, and the other choices must be then voltage gated, right? So the change in voltage is what would make the sarcoplasmic reticulum release the calcium.
Okay, so acetylcholine will first bind the ligand, and this will cause the sodium influx, and this will cause the calcium release. Okay, is
Yeah,
that the order? Okay.
yeah. Like, you know, yeah. So okay, you can so yeah, pretty much that. But I guess you can also have the reverse. Well, I shouldn't say that. I was thinking about like how the calcium. Here, let me try looking up calcium neuromuscular, because I'm pretty sure that it requires the calcium like to even start that.
Yeah, I see, like the neurotransmitter vesicle release is maybe what you're thinking.
Mm-hmm.
Mhm.
Yeah. So
Like in this case, the calcium is not released because of like a, you know, to exocytose something. It's more of, I think, the sarcoplasmic reticulum's function because it's storing a bunch of calcium.
yeah, yeah. So I guess calcium that can be present already can contribute to the release of um okay, so I think like it'll be like this. It'll be the axon terminal will when it when the action potential reaches that, it'll cause voltage gated calcium channels to open um uh in the sarcoplasmic reticulum, which would then have it come out into the neuromuscular junction, and then it can get taken in by the motor neuron. And when that's taken in, it will cause acetylcholine to be released, and then acetylcholine is released, binds to a receptor on the uh, motor end plate, and that opens up the sodium channels, allowing that influx to happen. So, so yeah, I would think about it um, in terms of like if they don't say anything about the channel, um, then your chances are it's going to be a voltage gated channel, but so that's going to be like most of the ones that we kind of like just talk about. But if it's ligand gated, it's going to require like a ligand to bind to some channel to open up that channel. And so if we just think about, all of these different types of examples, B is just talking about regular action potential generate propagation with the sodium influx, and that's voltage gated. And then the A and D, I guess, are going in opposite directions.
Yeah.
So, yeah. So basically, you can have all of these happen through that will be mediated by voltage gated channels, but the only ligand gated channel in this type of neuromuscular junction area will be um, uh, the acetylcholine binding to the ligand gated channel.
Let me just see.
Yep. So let's see what they say up top first. Okay. Um, all right, gated ion channels bind a ligand and open a channel through the membrane that allows specific ions to pass through. In contrast, while ligand gated ion channels rely on ligand binding to open, voltage gated channels open in response to a change in membrane potential. We're focused on the ligand gated one and not the voltage gated one. And typically, ligand gated channels bind a ligand like a neurotransmitter, so that can also help. And open a channel through a membrane that allows specific ions to pass through. It's going to be sodium if we want depolarization. A big one we see is in electrically excitable cells, like neurons, which need to react very quickly to a stimulus. When a gated ion channel opens, it rapidly lets ions move through the channel, either into or out of. Okay, so all right, and then they say for A, this is the distinction we're careful to make in the breakdown of the question. This is mediated by a voltage-gated ion channel, not ligand-gated. So, uh, so a change in voltage, aka like an action potential reaching the muscle, will cause that calcium to be released from the sarcoplasmic reticulum. And then B is just your regular run-of-the-mill, depolarization, essentially. in neurons, right?
Yep.
Yeah, and then so for this one, they're saying that yes, the acetylcholine is that neurotransmitter that will bind to the ligand-gated sodium channels of the motor end plate, and that's what requires a ligand-gated channel rather than a voltage-gated channel.
Yep.
So maybe like some context might help. So there's, well, do you know like what sarin gas is?
No.
Okay, so uh so it's funny because I actually learned this from this when I was a kid. There was this movie called The Rock, uh, which is about a deadlier form of sarin gas that's called VX gas, and these are nerve agents, which mean that they are acetylcholine esterase inhibitors. So we have our neuromuscular junction, and we know that acetylcholine can bind here and cause sodium influx, which can then lead to muscle contraction, right? Now, we're not going to have all of that acetylcholine, working, right? Some of it will be extra, right? And so some of the extra acetylcholine will be broken down by acetylcholinesterase, and that will help it get taken back in to that to the what is it called, the motor neuron, right?
Yeah.
So if you were to invent an acetylcholinesterase inhibitor, you are not allowing acetylcholine to break down, and you're not allowing it to be reuptaken into that motor neuron. So you essentially just have an excess of acetylcholine in the neuromuscular junction, which attaches to the ligand-gated channel, causing that influx, causing action potential propagation, which is leading to contraction of the muscle. And so what happens in this type of nerve agent is that um, that amount of acetylcholine is always there, so your muscles contract, relax, like spasmodically, all throughout your body, even like, the like muscles that are not what's the word, that are like involuntary, like your diaphragm and things like that. So basically it just causes like tons and tons of like twitching, like contraction. So that's how those things work. So so basically, um, at the neuromuscular junction, that is when you will see ligand-gated channels, because that's where neurotransmitters are required. Another example could be for like like parasympathetic nervous system will predominantly use acetylcholine as a neurotransmitter, but the sympathetic nervous system, do you know what that would use?
The sympathetic nervous system, glutamate,
So, so I know you're
like norepinephrine as well.
yes, yeah. Because like the glutamine, GABA, things like that are going to be more so like neurotransmitters that are like in your brain. But the ones that connect like, to like the muscles and stuff could be parasympathetic with acetylcholine, and then it could also be sympathetic, which will be the epinephrine, norepinephrine. Yeah. So those would also be ligand gated channels, right?
Mm-hmm.
So yeah, so think about like just how the vast majority of the time, if they just say that, because like when they say, yeah, so essentially just think of the neuromuscular junction as the one
I
that,
think,
yep.
yeah, I mean, I think I am clear on like the difference between like ligand gated and voltage gated. It was like the answer options were very
Mhm.
like specific. I guess I just don't know the pinpointing of like
Yep.
what exactly and when is happening.
Yeah, yeah, yeah. Yeah.
Yeah,
Yeah, it's going to be the at the neuromuscular junction. Yeah.
yeah. All right.
Mhm.
I guess what else? Could I ask you? Um, yeah. Um, yeah, this was no. I think I reviewed these. These should be fine. Yeah, I guess I'm like nervous with
Mhm.
like all the material. Yeah. If I were to do practice questions, I have like all the AMT material. What would you recommend like doing?
Uh, I think you've done like what you should be doing in the section bank, maybe the psych soc section bank to finish it up, but I also think that doing UWorld like this can help you out, because it would be kind of like drilling you on these kind of like difficult kind of concepts and stuff, and can also incorporate like you like reading the passage and things like that. But I think for the most part, you're in a good place. You know, uh, in terms of the practice tests that you've done, the questions that you're asking me here, and the I guess just the overall like yeah, like I would say like you're prepared, you're used to taking these tests. So I would so okay, let's see. So on your test on Saturday, right?
Yeah,
So and what, it's like 8 a.m. or something.
yeah.
So how's your sleep been?
Um, it's I've been trying to switch my schedule for a long time, but the time change, daylight savings really messed it up. I feel like yeah, I've been waking up around 6:30, so I think I'm good.
When
Yeah.
do you like go to bed?
Oh, 11 max.
Okay. How long do you feel like it takes you a while to fall asleep?
Depends how anxious I am.
Yeah, exactly, right? So that's the case. So like you want to like go to bed maybe like if you're finding that you're falling asleep like 2 hours after you go to bed, then we want you to go to bed 2 hours early just to make up for that. I mean, to do that like on Friday night, definitely. Um, so I would say, let's see. Okay, so I would say that um to maybe go through the psych soc section bank. Um, and yeah, try that out. And if we meet tomorrow, um, we can go over like some of those last minute kind of questions that can be either from the psych soc or just general, I guess. If you notice any type of, maybe mistake that you're making or any type of you know, like if there's a passage that you want me to kind of like help you go over, we can go over that stuff tomorrow. But that'll be for Thursday. For Friday, you just want to kind of like relax a bit, like not necessarily relax, but you don't want to do anything that would really cause you to fatigue yourself. So that would just essentially be like as much Anki as possible. Um, so so yeah, I would say so yeah, how do you feel about that? Because like, you were talking about doing like the section banks. Like were you planning on doing like the psych social one again?
Um, Not really. I was more of leaning towards doing the Chem Phys one if I were to.
Oh, I thought you did that from. Oh,
No, I did biochem.
got it, got it, got it. Okay, okay. So, all right. So yeah, maybe do that.
Or like half and half. I could do that. Yeah.
Yeah. I would say to do that. If psych social, like you really feel confident in it, then I think the Anki would be enough. But also like from those tests that you took from Blueprint, right? Any choice that you see, any term that you see that you don't know, to make like a card for it if you want, or like just to remember that stuff. But yeah, I would say that I guess your next thing to do would be perhaps like the CP section bank, Anki, like including the psych soc stuff,
Yeah.
and yeah, like yeah, and then whatever other questions you have for me, we can talk about tomorrow.
Yeah.
And then for Friday, you just want to kind of like rest, a little bit.
Okay. Sounds good.
Yep.
Yeah. Yeah. I mean, it's stressing me just the sheer amount of like Anki cards that like I haven't done
Oh yeah,
or
yeah.
see.
Yeah, don't worry
Um,
about finishing them. Like no one's going to really finish them, and it's not even like important to finish them, because if you finish them and you don't review them, like you're gonna forget it. The entire point of Anki is just to have things that help you remember stuff.
yeah.
So so just think of Anki in terms of like the time that you're putting into Anki rather than the cards that you're getting going over. Because yeah, the entire point of it is just to get stuff in your, long-term memory. So don't worry about finishing those decks. Just worry about learning, the decks that you see. So yeah.
All right.
Mhm.
All right. Sounds good. I think I'm going to try to wind down a bit.
Yeah. Even try to maybe like tonight or something, like go to bed maybe like an hour early or something like that, because it's just gonna take longer for you to fall asleep, right?
Yeah.
So it would be good to practice that. So tomorrow, do you want to do like I am only free tomorrow at 7 again? Is that okay?
Let's do it. Yeah.
Okay. And then that'll be like kind of like a last session. Like we could even spend that kind of doing maybe like, well, we'll see. We'll see it. But I was thinking of like maybe like even like a little test simulation or something. But yeah, whatever we do tomorrow, we'll you know
Yeah.
we'll complete like everything. So yeah, if you have some questions ready, that'll be good too. Uh,
Yeah.
but yeah,
Sounds good.
yep. But yeah,
Let's do that. I like that idea of like a test simulation.
all right.
I'm not sure what that entails, but sure. Yeah.
Okay, we can do that. So all right then. So yeah, try to go to bed earlier, and then I'll see you tomorrow at 7, right?
Okay, sounds good. Yeah.
Okay, awesome. All right. Yeah, keep up the good work, okay?
Thank you.
Thank you.
Bye.